The nanodispenser-based transfection protocol and developed tool described in this video represents the first user-friendly high-throughput transfection method, allowing even beginners in the field to be successful. This easy and high-throughput technique allows transfection of cells with 384 independent conditions. Furthermore, the method is low cost due to the nano volume of reagent needed.
To define the best transfection parameters for other cell types, we suggest keeping the source DNA concentration constant and using varying amounts of transfected DNA, transfection reagent, and number of cells.
For dispensing the 40 microliter cell suspension, mount a 10 microliter cassette on the peristaltic liquid handler device and prepare an appropriate program. First, adjust the flow rate parameter to low to dispense cells with a low speed to avoid promoting potential damage to the cells by shear stress and high impact on the bottom of the wells. Then, adjust the dispensation height to 11.43 millimeters. The height must be high enough to lower the cell impact on the bottom of the wells during the dispensing process, but low enough to avoid retention of the droplets on the dispensing head. Next, adjust the plate clear height to 16 millimeters to allow free displacement of the dispensing head over the plate after dispensing each row. Visually control the proper settings of the peristaltic liquid handler head height. Make sure no drops are retained on the dispensing tips while dispensing. Also verify that the head is high enough to allow displacement of the head after dispensing each row. To generate pick lists to drive the ADE